Okay, in the first video we talked about how we can translate proteins, have them fold in the cytoplasm, and then go off to various places. Now we want to talk about what happens if that's not where we're headed. What if we need to go into the membrane? Or what if we need to be transported off to another organelle or secreted out of the cell? All of those processes are going to go through this other option, which means we need to know this right away. We can't finish synthesizing the protein because it has to get over here and get synthesized at the surface of an endoplasmic reticulum so that it can then be sorted, modified, and sent off through vesicles to wherever its destination is. So that's the process we want to talk about today, is this vesicular transport and how that has to start right at the first step, just as the protein is beginning to emerge from the ribosome in the cytoplasm. So entry into this pathway, the vesicular transport pathway, means we have to have a different signal. Now you don't need to know what these signals are, don't memorize them or anything. You just need to know that a protein, as it's emerging from the ribosome, starts saying things that say, hey, I don't belong in the cytoplasm. Who notices that? There's a recognition particle, the signal recognition particle that says, aha, this guy is a protein that can't be synthesized here in the cytoplasm. We need to drag it off to the endoplasmic reticulum. Stop translating this guy. I want to make the analogy to a different transport system, one that you're more familiar with possibly. That is traveling to another place. You may be downtown and you're going to take a bus someplace, or you may be going to the airport. What happens when you get to the airport is you need to go through some recognition system. So the security system at the airport says, okay, show me your ticket, show me your ID, show me that you belong here, then I will let you through this system, through this security gate, and now you can go off to the gate where your airplane is. So your airplane's sitting over here. You don't have to go through security again to get onto the airplane, you just have to show your ticket. The airplane now flies you off to Atlanta, and you come to another gate. You can get off the airplane. You don't have to show your ticket or go through security again, but you travel inside the system. Now, Atlanta has this very large airport, so you might have to get onto a train and take a train to another place. You still don't have to go through security. Now, you get on another airplane and it flies you off to Madrid or wherever you're headed to. You still have shown your ticket, but you haven't gone through security again. Now, how do you know where to go? Well, we're taking everybody who's going to the same place when we're bringing them to this gate. We say, okay, everybody who's going to Atlanta, go to gate D11. Get on the plane to Atlanta. Everybody who's in Atlanta and now is going to Madrid needs to make their way to the A concourse, get on that plane, and go off. So we're going to collect everybody who's going to the same place. We're going to make sure they belong there. And then we're going to transport them off in these vesicles or airplanes off to the place where they're going. And then when they finally get there, they can exit this secure bubble. But that security system travels with them all along. The analogy to that is you only have to cross the membrane once you've entered the vesicular trafficking. The inside remains the inside by a series of fusions and openings, but we maintain the security of the inside of the system. And we only release this at the very end of the process. So we're going to collect things going to the same place, we're going to make sure that they belong there, and then we're going to take them in smaller transport vesicles off to the place where they're going. And here's what that looks like inside of a cell. As we were saying, we've got this ribosome. We started to make a bit of the protein. The protein was recognized by this signal recognition particle, the SRP, and said, wait a minute, you're the thing that can't be made in the cytoplasm. I need to stop the translation, drag you over here, engage a pore in the endoplasmic reticulum, release that so that now the protein can go inside as it's being synthesized. Now I'm going to release the ribosome and tell it to continue translating, and the protein will go through this pore and into the lumen of the endoplasmic reticulum, just like going into the airport. We'll worry about which gate you're going to go to after you've gone through this security system. one important variation on that theme is, suppose you're supposed to be in the membrane. Well, here I don't want to force you through the membrane and into the lumen, where you're going to have to try to fold and be soluble when you're not a soluble protein. So instead, I'm going to have yet another signal. So I had this signal recognition particle that said, take me to the endoplasmic reticulum. Now there's another one, this stop transfer sequence that says, this looks like a membrane protein. Why don't you just open up this pore and a little slit in the side and let me out into the membrane, and then you can finish translating. So I'll have part of me inside, the N terminus inside, and the C terminus outside. And those guys can try to fold up independently. But this is the world that I live in. I live partially in the lumen and partially in the cytoplasm. This is going to become important because we're going to modify stuff in here in the lumen of both the ER and the Golgi, and that's going to end up being the outside of the cell. And that provides at least part of the answer to the question, how do cells recognize each other? The answer is, we modify the proteins as they're being sent out to the surface with a variety of very complicated sugars that we do not need to get into. N-linked and O-linked glycosylation just means I took sugars in various configurations. Here's one, glucose, 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 and a mannose, and some other modified sugars. We don't need to worry about them. I just want you to know that there's an incredibly bizarre series of glycosylations that can happen, and they can go on to either a nitrogen or an oxygen of these proteins as they're coming into the endoplasmic reticulum. So the first thing that happens is a protein comes in and it gets an N-linked glycosylation. Here's your sugar basket. Welcome to the endoplasmic reticulum. Thank you very much. This is also going to be used to monitor whether or not folding is going properly. But ultimately, these N-linked glycosylations and O-linked glycosylations are going to end up being the outside of the cell. So the inside of the lumen of the ER and the Golgi and the vesicles turns out to be the outside of the cell. And we'll look at that again in a minute. Okay, so bottom line, there are these strange modifications that happen. These sugar modifications that happen as you go into the ER lumen, and those turn out to make proteins have a distinct personality, and that makes cells have a distinct personality as these proteins make their way to the surface if they are membrane proteins. All right, and we sort of skipped over the magic step of folding of proteins when it happened in the cytoplasm. We mentioned that there were chaperones. This is even more important here in the endoplasmic reticulum because we're not going to let anything go on to the next stage into the Golgi apparatus until it has demonstrated that it can fold. Again, folding is very difficult. Most proteins actually that start getting synthesized don't end up folding properly. It is the most common outcome. So folding is a very difficult thing, and it needs these guys who are chaperones, who are going to help, guys like Calnexin down here. They don't know what a folded protein looks like, but they know that hydrophobic patches are bad. It shouldn't be hydrophobic patches at the surface. So if they find hydrophobic patches, they bind to them, and they just squeeze, literally squeeze them, and then say, try it again. And they give it a few tries, and if it doesn't work, they say, you can't be folded. You're going to have to exit from the ER, and we'll talk about that in a minute. If they do manage to fold, then they get part of their glycosylation changed, so that gets removed and modified, or partially modified, partially removed, and sent on. So this is sort of a scorecard and a modification of the protein that says, I've been trying to fold you, and you either have or haven't worked. So these chaperones are both attempting to help, and they are keeping score of how many times it has been tried. If this goes on too long, the chaperone over here, still bound to the hydrophobic misfolded protein, says, let's go over here. I've got a friend that's going to help you out. It goes back to a pore in the ER membrane and sends the protein out. That now gets signaled to have these ubiquitins added to it. Ubiquitin is a small 8 kilodalton protein, and it is a signal that this protein should be degraded. So the chaperone was lying. He wasn't really going to help him. He was going to help him in the sense that he's going to get recycled. But really the next thing is into the proteasome you go, and you come out as little chewed up bits. We'll talk about the proteasome quite a bit later when we talk about cell cycle issues, because this is a convenient way to get rid of proteins in a targeted way. In this case, they're targeted because they're unfolded, but we'll have other proteins that are perfectly fine that we want to get rid of because we don't want them anymore. And the proteasome is going to be the answer to that again. All right, so if you don't get folded properly, you get sent out and degraded. If you do get folded properly, you get to go on from the endoplasmic reticulum into the Golgi body. And the way that we get there is the first of the vesicular trafficking steps. So here we are in a donor compartment, and we're sitting all together here like in the concourse at the airport, and we have to get sorted out. We say, okay, who's ready to go to the Golgi apparatus? I am. So all these guys have to get together over here and get packaged into a vesicle. The vesicle then buds out, and the process is well understood. There are these proteins that say, okay, I'm a cargo. Everybody who's going to the next step, come with me. So it binds to the cargo, and then there's these proteins called clathrins that make the membrane start to bud. We don't need to know anything about that. I just wanted you to know that it is an active process. Somebody's coming along, making a bend in the membrane, and then it'll pinch off and send off this vesicle, this little membrane-bound compartment with all the cargo inside, with everybody who's going to the same place. So the cargoes have to have been identified before they got in here, and we also have to put something on the outside that says, and here's where we're going. We're all going together to the same place. Where is that place? So this vesicle gets sent off with signals on it, and it will now fuse with the target compartment, wherever that is, and now the stuff that was inside the lumen of the ER is inside the lumen of the vesicle, is inside the lumen of the target compartment, all without having gone through security again. No, they didn't have to pass through a membrane. It's just being pinched off as the soluble stuff and sent off to the next place. And one set of targets is going to be the Golgi apparatus. So we go from the ER and we go into one Golgi stack and we go to the next Golgi stack. We get modified some more. We go to the next Golgi stack. Eventually we're done. We're finished with this, and we need to go to where we were headed to in the first place, the lysosome or the plasma membrane or wherever it is we're going. So we get everybody together who's going to that same place. Again, we get off into one of these little vesicles and we head off to where we're going. We might then fuse with the plasma membrane, or we might fuse with the lysosome. Wherever we're going, we all go there together. And now the inside of the vesicle becomes the outside of the cell, or the inside of the lysosome, wherever it is we're going. And here I'm just pointing out that endocytosis is just the opposite of this. We might have a regulated secretory pathway, say a neurotransmitter, and we're going to sit here at the membrane and not release until we want to, until we're told to. Or we have these constitutive secretory pathways where we just always, as soon as we get to the plasma membrane, we fuse and send our stuff out, leave our membrane proteins in the membrane, and send our contents out. So this is constitutive, this one's regulated. And we can just reverse the process by bringing stuff in. We'll pinch in and then send stuff into the endosome, and we'll look at that in a minute. We pointed out earlier that sometimes this is more complicated. Not all cells are tiny little guys that are microscopic. Some of them are a meter long, like neurons. So we need to have a way to get along this meter-long thing, and so we can't just depend on passive diffusion for everything to happen. So we have this active process of transport that'll go along microtubules. So we take this microtubule, this track, and we take these motor proteins, these dyneins, and we link the vesicle to it, and we say, okay, come with me, we're going down, we got a meter to go, we're going to go down to the end of this neuron, and maybe you'll be released there. This is going to be important as we talk about things like peripheral neuropathy. If we destabilize the microtubules, for reasons that will become obvious later as we try to prevent cells from making microtubules to go through mitosis, we're also going to have the secondary effect of making this microtubule-based transport process inefficient, and we'll end up with peripheral neuropathies because we won't be moving these vesicles down to where they need to be. So this active transport also goes on, but only in very large cells. All right, we'll call this slide, "Are we there yet?" How do we know when we're there? How do we know when we've reached the membrane that we're supposed to fuse with? So we have to have signals that were incorporated. Here's the clathrin-coated pit turning into a vesicle. We also incorporated a snare. I'm not going to get into what snare means because I like it better the way it is. We have vesicular snares that are going to sit in the surface of the vesicle, and we have target snares that are going to sit in the membrane of the target. And if these two guys like each other, the light blue and the light red like each other, they will snare each other, stick together, cause the membranes to fuse, and the stuff that was inside will go inside that target. That won't happen with just any old target membrane. They have to be matched. So here's a bright red one and a darker blue one. Those guys get together. So we have to recognize the appropriate snare combination before we will fuse. So that's how we're going to recognize our target, as we have a series of different snares, and we incorporate them both into the vesicle and we make them on the target, so that we know that those guys are belonging together. And of course, everything gets incredibly complicated, so we don't need to get into the details. Just think about it like this. Here's our airport, the endoplasmic reticulum. We got into security. We're headed out. Then we're going to go into the Golgi, get modified, sorted, and then sent into these terminal vesicles that are going to go someplace and do something. Either fuse with the plasma membrane constitutively, just always being swept out there, or in a regulated way, like neurotransmitters, and the reverse process with different kinds of clathrins. Same family is going to just be the opposite, where we're going to bring things into the cell, bring it to the late endosome, early endosome, late endosome, and then do whatever it is we were doing with things that we were taking in from outside the cell. Important thing to note is that if things go to the wrong compartment, they end up in the wrong place. This is going to be a useful diagnostic trick, because if things end up out here in the extracellular environment, then they're going to end up in the serum. And we're going to use tests of serum to say, okay, is everything going to the right place? With cystic fibrosis in particular, we're going to ask, is there immunoreactive trypsinogen, something that should be in the lysosome of particular cells and should be getting sent into the digestive tract? If it's not going to the right place, it'll end up in the serum, and we'll use that as a test to see if cystic fibrosis is occurring in a patient. So if things go to the wrong place, we can catch them there through various diagnostic methods, and we'll talk about those during the TBL itself. All right, so hopefully this looks a little bit less intimidating now. We've gone through each of the steps. We're going to go transcription, translation, make protein. The protein either gets made in the cytoplasm and then used locally, sent back into the nucleus, sent off to the mitochondria, or it has to have gone through this other branch and go through the vesicular trafficking, where the translation is halted, it's brought to the endoplasmic reticulum, released for more translation, or put into the membrane, like CFTR itself will go into the membrane. We're going to do a bunch of modifications here in the lumen, and that's going to affect parts of membrane proteins or all of the soluble proteins. We'll have vesicular trafficking that brings us to the Golgi, more modifications, and then we either have regulated secretion, where we're going to wait, get a signal, and then go out of the cell, or we're going to have constitutive secretion, which is the default state. If you aren't told to do the regulated form, you do this default form. And very important here, the stuff that's sent out, secreted proteins, or the membrane proteins, have these modifications that they got on the inside of the ER and Golgi are now presented to the world as the outside of the cell, because this is the final inversion of configuration, where we now have the lumen of the vesicle as the outside of the cell. And that means that each cell can have a personality that was dictated by what happened during this vesicular trafficking. We can also send things off to the lysosome. The lysosome is then going to be responsible for turning over old mitochondria and for fusing with endosomes to chew up the stuff that came in through endocytosis. If things don't go to the lysosome, you can imagine that these processes are going to fail. So we have to get everybody to the right place to do their function properly, and this is the system that we use.